Hello, we are Colony 5, the Bamboos. My name is Brian.
My name is Annika.
My name's Brian.
And my name is Mary.
And we will be presenting on the topic of engineering organisms to solve difficult computational problems.
Okay, so you might be asking why we should use organisms to solve these difficult computational problems. The answer is simple. Organisms like bacteria and mold have various properties that can be useful if applied computationally. For example, bacteria can replicate extremely fast and inexpensively, and certain species of mold are able to search for optimal routes among food sources. Another interesting reason to use organisms to solve problems is actually because of their DNA. DNA can be used to build supercomputers that can solve difficult computational problems. This is for three reasons. DNA's ability to hold large amounts of data in a small size, their help in increasing performance rate, and parallel computing. Because of this, DNA strands can actually replace the traditional hard disks in computers. The truly amazing part is that all the computations are almost completely parallel. You essentially have thousands of DNA strands replicating and forming independently of each other. Okay, so one way that organism can be engineered to solve difficult computational problems is through DNA computing. DNA computing is an emerging branch of computing that uses DNA and molecular biology hardware instead of traditional silicon chips. This idea was founded in 1999 by computer scientist Leonard Adleman. There are many differences between DNA computing and normal computing done by laptops, computers, and other devices today. Instead of using silicon chips to send/receive signals and data, DNA computing uses biological molecules, namely the nucleotides A, C, T, and Gs of DNA. On the other hand, computing devices today use binary to operate, which are combinations of zeros and ones. Finally, DNA computing operations and computation is done by splicing, aligning, and copying DNA to get a final product, while normal computing uses electrical switches. In DNA computing, an algorithm's input is represented as a sequence of DNA. Instructions are then carried out by lab procedures on DNA, like gene editing. The result, which is usually the answer to the computational problem, is some property of the final DNA molecule. Okay, so Brian L. will go deeper into the DNA computing specifics.
So one important part of computing today is logic gates, and logic gates are essential to programming as they can process inputs and return a true or false outcome. And there are many possible logic gates, including the popular OR gate and the AND gate, which we've used many times throughout this program. The OR gate returns true if one or more of the inputs are true, and the AND gate only returns true if both inputs are true. This can be represented by DNA computing as well, by using complementary sequences with a fluorescing or glowing molecule attached if it binds to another sequence. When you input two DNA strands, they will bind and glow to the complementary sequences, and if they're complementary. The fluorescent molecule can also be modified so that it will only glow if both strings bind, or it will glow if one string binds. We can infer a true or false outcome based on if the DNA glows or not, which is representing a logic gate. So now we'll move on to one of the first computational problems that we'll be solving using DNA computing, which is the famous Hamiltonian Path problem. So this problem involves finding a route between 7 towns, and each town can only be visited exactly once, and you can only start and end at a specific town. So you start at a specific start town and end at a specific end town. So we can do this by setting the towns as random 20-mer DNA sequences, which is what Leonard Adleman, the first person to solve this problem, did. Adleman also set paths between the towns represented as 10-mer DNA sequences. And he mixed these 10-mer and 20-mer town and path sequences many times and ended up with many different sequences of towns represented by DNA strands. So how does this work? As I said before, each town is represented by a DNA strand, and the path that connects these two towns is half complementary to each town includes. For example, in the image, vertex 0 is representing town 0, for example, and vertex 3 represents town 3. And here we have an edge 0 to 3, which is the path between vertex 0 and vertex 3. As you can see, half of the edge is complementary to vertex 0, and the other half is complementary to vertex 3. And when they bind, it will allow vertex 0 and vertex 3 to attach to each other. And this is how the towns are attached. And if you do it many times, you'll get many different combinations of towns. And now we have to move on to trimming these sequences, because not all these sequences are correct sequences. So the first step is to run PCR, which is polymerase chain reaction, to amplify the sequences that are starting and ending at the right town only. So most of our DNA will now be made up of sequences that have the right start and end time. Step 2 is to run gel electrophoresis to separate the remaining strands by length. So we make sure we don't have strands that are way too long because there's only one possible length. So we'll only take the ones with the right length. And step 3 is to remove sequences with repeated towns through affinity purification, which removes any towns that repeat. For example, you can't go to the same town twice as shown in the instructions. So thus, after these 3 trimming processes, we're able to get many different possible sequences that fit the requirements of the problem. And this is how Adelman solved this problem. So next, Mary will go over another computational problem called the traveling salesman problem.
Similar to the Hamiltonian Path problem, the traveling salesman problem requires finding a route to every city on a map that also has a specific city of origin. However, in the traveling salesman problem, the goal is to find the shortest possible route given the distance between the cities, and the solution must return to the city of origin. One of the difficulties in solving this problem is that as the number of cities increase, the number of possible routes also increase exponentially. In order to solve this problem, researchers engineered a slime mold called Plasmodium because it can be repelled by light. Food for the mold was used to mark the cities, and light was used to guide and set the paths. Naturally, the slime mold wants to find the shortest possible route to get the food in order to conserve energy, thus solving the problem in a manner in which the number of cities can be altered. Upon observation, the resulting routes of the slime mold were extremely similar to the paths formed by computer algorithms, showing that organisms, in this case Plasmodium, can be used to solve difficult computational problems. Moving on, Brian will be talking about the pancake problem.
So the pancake problem is another problem that was solved using engineered organisms. And the goal of the pancake problem is to start with a randomized stack of pancakes and sort them with the minimum number of flips. Sets of the pancakes are flipped until the whole stack is sorted. In the image, you can see that after the flip, the order of the top 3 pancakes in the stack are reversed with the largest pancake on top. The burnt pancake problem is a variation of the pancake problem, where one side of the pancakes are burnt and the pile has to end up sorted with all the burned side facing down, unlike the regular version where the direction didn't matter. Researchers replicated the burnt pancake problem in bacteria by sorting DNA fragments using DNA inversion, and when the strand of DNA was sorted, it would result in the bacteria becoming antibiotic resistant. Unfortunately, there are some limitations. Bacteria are so good at DNA inversion that the inversion process had to be slowed down in order to allow for better control over the inversion and the recombination. And it's also only a proof of concept demonstration, showing that the DNA computing can physically happen in a lab. As the speed of inversion increases, the accuracy decreases and more mistakes in combining and splicing DNA are possible. Also, DNA in general is prone to transcription errors, whereas computers are more accurate. DNA computing can also use a lot of resources, and some problems are faster and more efficient on a conventional computer due to the length of time required to conduct the biochemical operations.
Even though DNA computing is currently slow, the size of the bacteria and the amount of data that can be stored in DNA means that it can be scaled up to a point in which millions of computations can happen simultaneously at a low cost.
So the field of DNA computing is an emerging concept that's still in its infancy, and its applications are still being understood. But there are big hopes for this field, and some of the future applications of this field are as follows. Nanotechnology. DNA computing seems to be the first example of true nanotechnology. If researchers are able to control the molecular devices, including DNA, inside every cell, they will be able to engineer devices more complicatedly and efficiently than current microelectromechanical systems. For example, DNA computers could be used to time-release medications, bolster organ functions, and provide medical feedback. Further potential applications might make use of the error rates and instability of DNA-based computation methods, such as a means of stimulating and predicting the emergent behavior of complex systems, and this could pertain to weather forecasting. DNA computing can also be harnessed to act along with the living cells to provide new detection methods in medical devices. And finally, DNA computers will be a cost-effective way to do DNA sequencing and would also not take as much time to sequence the DNA of humans and animals as it does right now.
These are our citations, and thank you for watching.
Thank you.